another generation. This is called gene family. The simple definition, the set of genes originated from one ancestor, only one ancestor. The first basic is only one ancestor by duplication process. That means actually for our convenience, our easy understanding, just assume here how many chromosomes here, 1, 2, 3, 4, 5. For example, 5 chromosomes. So these 5 chromosomes duplicated. First, these are duplicated means one chromosome becomes doubles. Like this, right? The single chromosome become double. This process called duplication. After duplication, it make it off and distributed into next generation. So for example, this is one chromosome. After duplication, it becomes 2. After separation of chromosome, become single.
One number.
Then this is distributed to the next. And same process again duplication process, again double, again make it off. Make it off means during division, such as mitotic division or meiotic division. Mitotic division are only in vegetative cell. Meiotic division before reproduction, before formation sperms, male gamete and female gamete. These are separated and enter into the gametes.
Yes.
And later, after formation gamete, these both gametes are fused. For example, I am telling you from one ancestor, just focused on 6 chromosomes. The 6 chromosomes. For example, this is XX or XY. Before gamete formation, this is duplicate. Duplicate means doubled. X. This is also duplicate. X and X. Here same. X, Y duplicate. Y. Now doubled. This doubled one make off and enter into the one gamete. This is enter into one gamete. This enter into one gamete. One gamete. What is fertilization? Male gamete.
Fusion of the male and female gamete.
Now these two unite. And entered into the another generation. So where they came from? Actually, they came from the very beginning ancestor. So one ancestor to these are carry forward to the further and further. It would be continued. So this is called gene family. And these genes are commonly the same biochemical function. Same biochemical function means because of same genes. For example, ancestor same gene code and next generation same gene code. So the biochemical function also same. Here the biochemical functions mainly carried out by proteins or protein enzymes. How the enzymes or proteins are developed or synthesized? Hello? Proteins or enzymes, synthesization. Already told you yesterday. Right. Transcription, translation. What is the basic? The substrate of the transcription first initially.
mRNA is required. DNA is required.
DNA. So DNA acts as a gene. If the same gene, for example, in ancestor, the same gene present, that carry forward the next generation. The what proteins are prepared in ancestor, the same proteins also prepared in offspring also, because the same genes. The same genes. Sometimes may occur the mutations also. I will tell you later What is the consequence of the mutation if any mutations occur in gene family? Before that, here the gene family mainly by duplication. Here the duplication two types. One is genome duplication, the mainly two types. One is auto polyploidization, auto polyploidization. Second one, allo polyploidization. Auto means same. The same genomes in a germ cell. No change. The same genome within the same genome in germ cell, duplication process takes place. That is called autoploidization. Second one, allo means the closely related genome. That means closely related species. For example, if we take the datura same species but variety different. Of course, the same genus and species, the small difference in variety are the
Brassica.
Right, Brassica oleracea also.
And variety capitata and variety another.
Brassica species, first one Brassica oleracea and another one?
Brassica oleracea only. Variety is capitata and another one is there. No?
Right,
capitata.
and next one?
I remember only one.
So anyway, closely related species. Mainly the duplication takes place in closely related species, that is allo. The same species auto. So anyway, you remember the gene family formed by duplication process, right? That is one important. And another important topic is tandem duplication. Tandem duplication. Tandem means, what is tandem? Repeated. Tandem means simply say repeated. The many gene families exist as a clusters. That means the gene is here also gene. The genes appears as a clusters. In tandem duplication, mainly here during the duplication process, two type of structural genes are formed. The tandem means repeated first here. This is one gene and repeated the same gene for our convenience. For example, this is globulin gene. Globulin gene. And it is for example, globulin gene, again the duplication, formation, globulin gene, again duplication, globulin gene, but sometimes this globulin separated into, that means forms two different type of genes. These two separate genes is alpha globulin gene and beta globulin gene. Alpha and beta alpha globulin and beta globulin gene. First, initially, this globulin found in, first the globulins are the proteins components of hemoglobins in mammals. These specialized globulin exist in embryonic or fetal circulation, but when the same baby reached the that mean maturity stage, in maturity stage the same gene, globulin gene, due to the repeated duplication, due to the small changes, due to small nucleotide changes, automatically it's developed into two separate genes. Of course, initially single gene and later two separate genes. One is alpha subunit and second one beta subunit. Right? After formation, this alpha and beta, now these changes can carry forward to further. That means in gene family, first one, almost what are the characters are represented or stored in ancestor, they can carry forward to further. At the same time, if any changes, if any mutations occur, they also carry forward. They
Yes.
also carry forward to further generation. That is the whole concept of the gene family. Right?
Okay.
Anyway, after formation alpha and beta, just the small structural, small functional changes, functional important changes. Where in globulin protein or globulin gene, they express the character in fetal, they mainly they capture the high oxygen because the baby, that mean in the initial state is baby inside the mother. They're not exposed to weather. They're not directly received by oxygen. They receive the oxygen received by from placenta, right? From
Yes, sir.
placenta. So they should need more affinity to absorb the oxygen. The capacity only have globulin gene. That's why inside the mother, the baby, the gene is globulin gene. That is capable of attract globulin from the placenta.
More oxygen. Okay.
Yes, more when compared with alpha and beta. When compared with alpha, beta means first one globulin. Later, due to the mutational changes, when reach the adult, after birth, maturity stage, it becomes hemoglobin. Hemoglobin, due to the small changes. The hemoglobin made up of alpha in it and beta in it. Alpha and Nishat. Nishat, this is the C8 plant molecular biology and biotechnology.
Okay, sir.
Unit number 4.
Thank you.
Okay.
Sir.
Right.
Okay, sir.
Right. So apart from the alpha and beta subunits, the further some pseudo genes also form. Pseudo genes almost non-functional. They are the small changes in during duplication. What is the duplication? Duplication means DNA copy duplicate, doubled with same sequence. If where the necessary necessary changes takes place, for example, globulin can change into hemoglobin. And apart from the hemoglobin, some of the pseudo genes also form due to the duplication. For
This
example,
globulin is nothing but our immunoglobulin. Sir, IgG, IgM, that is only.
not yes, immunoglobin.
Okay.
Immunoglobin. Right? For this convert into hemoglobin and further alpha, beta, and finally pseudogenes. So this is the small changes inside the genes. So these character also carry forward from the one generation to the another generation. Right? This is the gene family. It is not an important one, but anyway, remember the gene family short answer. This is short answer. Okay? Right? And next one, repetitive DNA. Repetitive DNA. Already told you about the repetitive DNA when I teach about the euchromatin and heterochromatin. Can you recall that? Anyone? Repetitive DNA.
It is having all the function nucleotides thick in nature is I think.
Nucleotide DNA. Sorry, repetitive DNA. Repetitive means repeated nitrogen bases. Simple. Repeated nitrogen base pairs. Then first of all, have you heard this word repeated DNA earlier? Already explained in the chromatin structure, euchromatin and heterochromatin. What is the difference between euchromatin and heterochromatin? Euchromatin. Okay, no problem. So euchromatin, mainly the repetitive DNA present in heterochromatin structure. Right? First of all, repeated DNA means the repeated base pairs. Repeated base pairs. That base pairs are, that mean adenine, guanine, thymine, cytosine.
Okay.
Such as the repeated sequence. So anyway, the first of all, in prokaryote, most of the genome codes only one type of DNA, mainly that is
BDNA.
non-repetitive DNA. Mainly non-repetitive DNA in prokaryote. Very less in repetitive DNA. For example, inside the cell own genome concept called as C value. This is the own this cell inside the cell, the chromosomes, chromosome genes. This set of an organism called as C value. In C value genes, the entire genes functional or active? No. This is functional and unfunctional also, active, inactive also. If you take the prokaryote, actually first before that, here the DNA two types. One is non-repetitive. One is non-repetitive, and second one repetitive. First, repetitive or repetitive. So of course, here in our book given REPETITIV, repetitive. So many may repetitive or repetitive. So first one, non-repetitive. In non-repetitive, actually this is non-repetitive. Another word is unique. That is the unique because this is the functional part of the genome. And this non-repetitive genome, non-repetitive means the nitrogen base is not repeated. The commonly continuous, for example, triplet codon, adenine, uracil, guanine, and guanine, cytosine, and thymine. It not be repeated. Guanine, cytosine, thymine again not repeated. So this sequence called as non-repetitive. For example, AUG, GCT, and ACG, like this. This is non-repetitive, and they are functional. That means these are involved in protein synthesis because this is the active one. And repetitive means the repetitive sequence not functional. In eukaryotic, this is this first one prokaryote. In eukaryote reverse. In eukaryote, 97% repetitive. Only 3% is the functional. Almost 97% repetitive means that is the junk, not involved in any protein synthesis. Of course, in C value, the entire gene, the only the functional active percentage in eukaryote only 3%. Only 3% active. Functional. Rest of the all junk file, not involved, not functional any synthesis of proteins and any biochemical activities. These are waste material, junk material in the form of tell me, one is repetitive DNA.
Introns.
Right, exactly. Introns and repetitive DNA, they are not functional. They should be removed. Only 3% active. Right? Let's see the types of repetitive DNA. First of all, concept clear. Repetitive DNA means the first, as a name given, the nitrogen bases repeatedly repeat.
Yes.
That's it. And mainly in eukaryotes, when compared with eukaryote and prokaryote, first in prokaryote, most of the DNA part is non-repetitive. That means most of the part is functional in prokaryote. Whereas in eukaryote, only 3% functional, non-repetitive, repetitive, and introns, that is unfunctional unit. Right? And let's see the types of repetitive DNA. Types of repetitive DNA, the mainly Types of repetitive DNA. Moderately repetitive DNA. Moderately DNA. Highly repeated DNA. Highly repeated. Highly repeated DNA. First moderately repeated. First difference moderately. First repeated clear. Right? Hello?
Yes, sir. Repeated clear.
Right. Repeated clear. Now moderately repeated means
Less
long
number.
nucleotide. Right. Long nucleotide base pairs with less number. Long nucleotide base pair with less number means, for example, this is the gene sequence. Guanine, cytosine, thymine in a DNA, adenine, cytosine, adenine, guanine. This is long nucleotide, not a short of almost in this more than 100 nitrogen bases. For example, 100 nitrogen bases length. The same sequence somewhere repeated. Same sequence somewhere repeated. But less in number. That means less than 10 number. For example, this is the entire gene. This is one of the entire DNA code, DNA strand. DNA strand here GCTACAG, same GCTACAG in a length, in the entire length, 2 times repeated. 2 times repeated. So repeated time is less, but nucleotide pair is long. Nucleotide pair long, but repeated time is less. That is called moderately repetitive DNA. Right? Next one, highly repetitive DNA. In highly repetitive DNA, the reversed. Base pair short, repeated more. Base pair short, repeated more. Let it complete with represent with blue color. The same DNA fragment length, entire length, whole genome length. The whole genome length, for example, here GC. Next, some of the common nucleotides, again GC, common again GC, common sequence
Cancer patient.
again GC, again. So this is guanine cytosine. This is repeated. This sequence is repeated, but short in base pair short. How many base pairs here in blue color? In each and
4.
each repeated
1,
sequence,
2, 3.
but in each and every repeated, how many nitrogen bases?
2 GC 2 are
Only two,
repeated.
only two nitrogen bases repeated, but more time. That is
Yes.
highly repetitive. Then what about the first red in color? Red in color, how many nitrogen bases for every repeat?
7.
7. So when compare with blue one, red one, nitrogen bases number,
2. Less.
the more in number. Nitrogen bases number more,
First
but
one is
repeated time
more.
less. That is moderately repetitive. The highly repetitive, less short in nitrogen bases, the repeated time more.
Yes.
Repeated time more. Given your textbook, the page number 66, clearly given. That is only 100 base pairs that are present in many thousands of time in the genome. 100
Yes,
base pairs, the present many thousands of time repeated. Right? So these are mainly highly repetitive DNA, mainly found in Telomere, already told you, telomere, centromere, and satellite DNA.
Yes.
Found in heterochromatin region and telomere, centromere, satellite DNA. Please, 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 please be silent. And this repetitive DNA not involved in protein synthesis. Not involved in protein synthesis. So this is moderate to and highly repeated DNA. And again, moderately repeated DNA, again two types, that is tandem repeat and interspersed repeat. First, tandem repeat. In tandem repeat, actually in book not given, just add it. In moderately repetitive. In moderately repetitive, after that, this again two types. Again two types. One is tandem repeat. Tandem repeat. Second one, interspersed. Interspersed. These two in moderately repeat. Moderately repeat. Actually not given textbook, just add these two. Tandem repeat and interspersed repeat. First one, tandem repeat. What is tandem? One after another. Repeated. One after another without gap. Initially we take from small gap, but in tandem, for example, this is the sequence. Here GC, again GC, again GC, again GC. So this repeated. So that means the nitrogen bases repeatedly without gap, continuously formed. That is the tandem. That is called tandem repeat. That means one after another. One after another, that is called tandem repeat. Again, this standard repeat. And next one, what is the interspersed? Interspersed means first from GC, later some nitrogen bases, again somewhere GC. That means it is interrupted. It is interrupted by some other nitrogen bases. That is interspersed repeated. Is it clear? Hello?
Yes, sir.
Right. So again, tandem repeat to three types: multiple copy, mini satellite, micro satellite. Is it clear? First one, moderately tandem and interspersed. Tandem without gap, one after another, repeatedly. GC, again GC, again GC. But here GC, but it is separated with some other nitrogen bases, again somewhere the GC repeated. That is interspersed. And again, in tandem repeat, again classified into three types. Have you noted this? Hello?
Yes, sir.
Make a notes. Please add this point. And first one, multiple copy. Multiple copy. Second one, mini satellite. Third one, micro satellite. First, multiple. The multiple copy called as mega satellite. The same as a name given, mega satellite, the repeated DNA sequence length is more. This is common limited to DNA length. The same repeated again. The same repeated again. So this is called mega satellite. This is called mega satellite. This is also tandem, one after another, one after another, right? The mini satellite, this is the medium nitrogen basis. Again repeated. So for example, here ACTAG, ACTAG, ACTAGC, something. Here only ACT, 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 repeated. The micro means very less in number. Very less in number repeated. Again AC, again AC, again AC, again AC. So this is micro. The mainly the mini satellite and microsatellite sequence mainly involved in DNA fingerprinting. These two, that means, for example, this repeated DNA, for example, the microsatellite, for example, microsatellite, a person, A is the person, B is the person, A person, this microsatellite repeated 100 times. In B person, this same microsatellite chrome repeated 500 times. So another person, another. So this is the individual, different in each and every person. So that's why this is the involved in fingerprinting identification, right? This is about repetitive DNA. Is it clear? Hello?
Yes, sir. Yes, sir.
Yes, sir.
Right. Right. Can I rough?
Yes,
Okay, sir.
Moderately tandem, in tandem, multiple copy, minisat multiple copy, or macro or mega satellite. Second one, mini satellite. Third one, micro satellite. That's it. The rest of the matter as it is in what given in our textbook, page number 66, right? Okay. Next 4.5, organization of eukaryotic genes. Repetitive DNA also short question. Repetitive DNA also short question. Actually, this repetitive DNA not involved in genome structure, but that means DNA, sorry, protein structure. So anyway, we discussed here as a part of the gene, right? Next, organization of eukaryote. This is very important. Organization of eukaryotic genes, right? Of course, the given the more matter, but we need only what are required for the transcription process. Only what portion involved in transcription, that only I focused. Okay, right.
Okay.
So anyway, organization means already some of the gene structure we completed, that is promoter and enhancer, silencer not completed. Anyway, promoters already completed, right? So in promoter genes, I will tell you what are the enzymes involved in protein synthesis. I will tell you later. But before that, gene structure. First, promoters. Mainly in genes, only 3 regions. Very, very important. Promoters.
Enhancer, starter box.
That is part of promoter, right? Enhancer.
Starter
And
box.
silencer. Silencer, right? These 3. And next one, structural. Structural genes. Structural genes. Is it clear first? These gene groups are clear?
Yes. Yes, sir.
Right. Right. For example, genome length. Sorry. Promoter region, enhancer region, somewhere this is silencer region, and this is structural region, and this is starting point, blue color start point. This is very, very important. If you have any doubt, ask me. The blue color line, starting point of transcription. This starting point, first one represent with plus, left side minus, 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 and plus continue this side, right side, right? On this region, left side plus left side region called as down, sorry, upstream. This is very, very important. Upstream, upstream, downstream, right side, downstream. Hello? Any
So,
disturbance?
No, sir.
No.
Any
Continue, sir.
disturbance? My side. My side. Any disturbance to
no,
you?
sir.
No, sir.
Okay.
No, sir.
Okay, right, right. So the middle starting point, this is upstream and this is downstream. Is it clear first?
Yes.
Right. In upstream region, mainly promoter first, next enhancer, silencer. Is it clear? Just
Sir.
wait. Promoter, enhancer,
Silencer.
silencer. Right? Okay. Upstream, downstream, clear? Right.
Yes, sir.
Right. Right, right, right. Please silent. Please silent. Right. Promoter, further enhancer, and silencer. And after promoter and operator region also. This is operator. That means operating gene structure. And next they are all structural. They are all structural. Somewhere here the start point. Start point. Upward. Downward. Right? Here bind RNA polymerase enzyme. I will tell you later how it is bind. Okay? Is it clear? Hello?
Okay.
Right. So now in promoter, already told you two regions. In promoter, two regions. The basal promoters are CCAAT box and Tata box. These two are the repeated motifs. Motifs means repeated sequence. The Tata box resides the 20 to 30 base pair upstream. Just wait. DNA length. Present term this region promoter region. This is starting point plus upstream downstream, and the promoter elements are CCAT and Tata box. The Tata box resides the 20-30 base pair. That means from this 1, 10, 20, 30, this region 20, 30, this region Tata box TATA. This is motify. This is motify sequence. Motify means repeated, and also TA. If you read any direction, TA 3 and to 5 and also 5 and to 3 and also this direction same, and this direction also same. This is called motify, right? In promoter region, blue color line, Tata box, and somewhere about 50, 80 base pair, 50 to 80 base pair or 70 base pair. Here CC AT sequence. CC80 sequence. This is one sequence. Tata box sequence. This is the two sequences important in promoter, because this site for implantation of RNA polymerase.
Okay.
Of course, for example, what are the requirement for synthesis of mRNA from DNA?
Template is required, primary is required.
Right. First one, template. Template, already told you that template is the DNA strand. The DNA strand is template. From the DNA strand, mRNA prepared. For preparing mRNA strand should require RNA polymerase enzyme.
And
RNA
ligases.
polymerase. In prokaryote, only one type of RNA polymerase. In prokaryote, one type of RNA polymerase. Whereas in eukaryote, 3 types of RNA polymerases are there. 3 types.
mRNA, tRNA, rRNA.
No. RNA polymerases enzymes. For preparation mRNA different, preparation tRNA different, preparation rRNA different. For example, here RNA polymerase 1. RNA polymerase 1. The RNA polymerase 1 for only synthesize rRNA, ribosomal RNA. Right? And 2. these two RNA 2, this is for mRNA synthesis, and 3 for what are the remaining tRNA synthesis. tRNA synthesis. So RNA polymerase 1, rRNA, RNA polymerase 2, mRNA, RNA polymerase 3, tRNA. So they are sequence. So anyway, for preparation, these rRNA, mRNA, tRNA, they are all. What is the template? These all. What is the template for RNA, rRNA? What is the template for mRNA, tRNA? The everyone the template is DNA, right? Right from the DNA strands, rRNA prepared, mRNA prepared, and tRNA prepared. First of all, to synthesize of any type of RNA required RNA polymerase. Now, this RNA polymerase, how it is attached, bind to the DNA strand. For how to recognize the appropriate position,
Locus is required.
that
Proper
locus, how it is identified. Of course, this
depending
entire
upon
length
the
unit.
required base pairs,
For
the arrangement.
example, right. For example, there are those specific sequence in a length of the genome. There are no specific. Then how the RNA polymerase identify the specific spot, specific place? Then how it is bind to the DNA strand. So here mainly to binding site, to initiation transcription should require some special sequence. The special sequence called as consequences sequence are modify. They are located at two regions in promoter.
Yes.
The two regions are that 20 to 30 base pairs 1 and 70 to 80 base pairs 2nd. These two, the specialized sequence. The specialized this sequence recognized by RNA polymerase 1 or 2 or 3. After recognized, then only bind to the specific site. Now the bind to the specific site, that means this promoter site bind with RNA polymerase. Now transcription started. Now after bind with polymerase, transcription started. It is very, very clear.
But before that, we must discuss about unwinding ligases.
No. First of all, after attached with RNA polymerase, after that unwind stand. First,
no, sir, there must be some opening so that you can bind, na, sir.
Where?
In DNA.
Okay.
For binding,
how many strands
binding the
right.
Two strands are there, na? They must get open, na, first. Then only this particular primer or everything is going to attach there. So for that, first opening is required. Fork should be formed. Replication fork. After formation of, first ligases plays a role there. So by means of the ligases, the fork, the replica fork is going to form. So there, this primer will come, it binds to the particular strand. So then the transcription takes place.
You are talking about the DNA replication. Of course, these both are same process.
This also opening is required, na? So though it is also, the DNA replication. Or...
Here also same process, but after binding RNA polymerase. Okay. As you said, what are requirement for unwinding?
Ligases.
Ligases not unwind. Ligases only dissolve the hydrogen bonds between
nitrogen bases.
Right. That is the ligase function. That means first, before that, give me one simple answer. In DNA, two strands. One is 5' to 3' and another one is 3' to 5'. Right? What is the template strand in both?
5' to 3' is the leading one, and 3' to 5' is lagging strand.
You completely confused with DNA replication and DNA transcription translation. First of all, your doubt is before binding RNA polymerase, the DNA strand should be removed, unwind, right?
Yes, yes, sir.
That is your right. Okay. As you said, after unwinding DNA, where RNA polymerase bind? Now here these two strand 1 and 2. As you said, this is unwind. Unwind means separated. These two separated, right?
Yes, sir.
Isn't it? Now
Yes.
this simple strand and this is also one strand. Then where the RNA polymerase bind?
A particular sequence which is
Where the particular
20
sequence? That means
this
where
is primer identifies that. This 20 base pairs one primer that identifies the loci.
Before that, I said Tata box, right?
The Tata box and CCAT box.
Tata, TA.
Yes.
Repeated sequence if any direction. Any direction means 5 and 2, 3 and 3 and 2, 5 and same direction, right?
Yes.
Okay. As
Okay,
you
sir.
said, it is if it is unwind. Where is Tata box? Can you found?
No.
Can you found
It
Tata
is
box
there
now?
on the primer. It is there on the primer now, sir. Sorry, it is there on the gene, which is having primer and all. And primer only, no, sir. Primer identify.
Okay, Parveen. Just wait. Let me clear. According to you, you are actually, for example, in DNA replication, this is DNA replication. In DNA replication also, after binding some of the requirements, then only this is unwind. This actually this initial after binding, one by one unwind. For example, this is this helix. This helix. While the polymerase, while these components move forward, this is unwind. This fold unwind. First one. Second, this fold unwind. Second, third, this fold unwind. Not at a time the DNA unwind. One by one.
Yes,
First one helix, second another, like this. After that, the new strands are synthesized. New strands are synthesized. For example, these two. This red color to hold one. Blue color, one direction. And another direction, blue color. Right? 1, 2. Now these are separated. These two are separated. That means one old, one new separated. One old and new separated. And they enter into the cell. That is DNA replication. But what happened in DNA transcription? In a DNA transcription, the same process. Right? This is DNA. In DNA, only one strand act as a template. That is, remember, you should remember only one strand template. In DNA replication, both strand act as a template. DNA replication, both strand act as a template. 3 and 2, 5 and template, and 5 and 2, 3 and also template. But here, only 3 and 2, 5 and is the template, not 5 and 2, 3 and. This is not a template. Right? Please be silent. Right. Actually, for our understanding, it is a linear shape, drawn as a linear shape, but is not a linear shape. You know, it is always scaled nature. DNA always scaled nature, right? Before unwind. Listen carefully. Parveen, are you Parveen?
Yes. No, right. I'm listening, sir.
Okay, Parveen. Right. In this DNA, actually, this is not an unwinding. Winding, only the winding in natural process, DNA winding, then only here polymerase bind at the place of Tata box. Promoter Tata box, because this is the recognition site. Now after that, while it is moving, that first step, initiation. First only initiation step. Initiation means only binding. After elongation, during elongation, as you said, the hydrogen bonds are cleaved. Hydrogen bonds are cleaved. And this RNA polymerase 2 move forward with help of some factors. While the RNA polymerase move, these chylings are unwind. These chylings are unwind when these RNA polymerase move towards one end to another end. Parveen? Hello?
Sir, listening.
First, this is RNA polymerase 2, right?
Okay.
Immediately not unwind the DNA. First of all, remember that word.
Okay, sir. Okay.
First of all, you should remember in transcription process 3 steps: initiation, elongation, termination. In initiation, the initial stage DNA in natural position, like a coiled position.
Okay.
After binding of initiation factor, also same. After elongation factors, gradually one by one helix deleted, separated. While the helix separated, the RNA polymerase to move forward.
Okay, sir, I understood.
At a time on the template stand 2, 3 and 2, 5 and 3 and 2, 5 and for our better understanding, I drawn here 3 are blue color. 3 RNA prepared. mRNA prepared. This mRNA stand prepared from 5 and to 3 end. This is important because the template stand 3 end. What is the nature of DNA? Antiparallel, right?
Yes, sir.
Antiparallel means 1 and 3 end, other end 5 end. So first initiation start is 3 end region. Automatically here the 5 end mRNA start. This mRNA continuation. అర్థం కావట్లే ఇంకా. Okay, Parveen?
Yes, sir.
Right, this is DNA. Polymerase, after binding, initiation factors also, after adding elongation factors, now immediately first this helix unwind. This helix unwind.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
This already unwind. Now,
sir, रुको इरशान.
Right, please.
My son is this.
Here mRNA prepared. Stop here to the second helix. Now second helix also unwind. Then what happened? mRNA stand prepared. Right? Is it clear, Parveen? Hello?
Sir, please have a look at my videos. I'm showing. Is it the one you're saying each helix is going to? Sir.
Okay.
Is it okay, sir? Ishan, Hatto. Sir, is the diagram okay, sir? This one only.
Where it is?
One opening of. Please see mine.
We will discuss later, Parveen.
Yeah, okay, sir. Sorry. Please carry on.
Okay. Right.
Yes, sir. Thank you.
Not Parveen. Anyone, if you have any doubt, please interact me later after the class. Okay.
I'm sorry, sir.
Okay. Right. So anyway, the gene structure, promoter, enhancer, and structure genes also completed, right? Any doubt in this? Hello?
No, sir. I have understood.
What about rest of the? Again, sure, just wait.
No doubt, sir.
Just wait. Just wait. Photo downloads. Right? Is it clear? Diagram clear to you?
Yes, sir, it's clear.
Right. Only I shown promoter site only. Only promoter site, right? Only promoter site. For our clear understanding, I shown the both prokaryotic site. This is prokaryotic promoter site. This is eukaryotic promoter site. Right. Eukaryotic, as I said, the Tata box region. This is Tata box. This is CAA TC box. So this region bind with RNA polymerase 2. Now +1. This is start region. We can observe here starting point also. This is plus. Starting site of the mRNA synthesis. Right. First of all, this is the DNA structure. Is it clear?
Yes, sir.
Mainly focus on promoter site. Right. And what is enhancer importance? Promoter completed. Promoter region only for initiation of transcription. That's it. That is the initiation site. Promoter region, initiation site, and binding of RNA polymerase. That is the site of the promoter. That means promoting transcription. The what about enhancer? Hello? Enhancer. What is the function of enhancer? The enhancer elements are required for maximal transcription to occur. The mainly this helps in control transcription. Here the gene promoter, the promoting transcription, this transcription process controlling by enhancer. Controlling by enhancer. So the enhancer gene sequence controlling promoter. Is it okay? Hello?
Yes, sir.
Right. They may be located the upstream of the 100 base pairs. We can observe in page number 68. Page number 68, the 100 base pair enhancers. Right. Next, silencer elements. Silencer completely repress the transcription. Silencer repress the transcription. Enhancer, as a name given, enhancer enhanced transcription and controlling transcription. That means, for example, after adding RNA polymerase at the site of promoter, it will be started with help of an enhancer mechanism only.
Okay.
This enhancer mechanism speed up the reaction and slow down the reaction only. But here the silencer's genome just site to the enhancer, the silencer elements, it is completely repress the transcription. Repress the transcription. Just wait, I will show the picture. Okay. This is not opened. I will show, just wait. Can you download? Right? Screen is clear. Hello?
Yes, sir.
Right. So this is the enhancer promoter. After binding with RNA polymerase, this enhancer activates this promoter region. Then only the RNA polymerase move forward while the unwinding the DNA strands, and it move forward. As a result, protein synthesis. That means mRNA strand prepared. The what about this is repressor? Silencer is repressor. This silencer, when silencer activate, act on promoter region, but even if attached with RNA polymerase, that not prepared mRNA strand, because the silencer control enhancer activity. Enhancer activity, that means repress. Silencer is the as a repressor, that is repressing the activity of preparation of mRNA and DNA strand. When required, mRNA strand automatically enhancer activated and promoting mRNA strand. Not required mRNA strand, automatically silencer activated and stop the activity of promoter.
Yes.
Right?
Yes.
So this is enhancer and silencer, promoter and structural genes of the RNA. Now, not explained about the structural genes. I will explain later about structural. While structural, nothing but what is the structural? Structural means
Chemical.
no. Your structural genes. What is the function of structural genes? Promoter genes, promoter region, promoting the transcription and binding RNA polymerase. Enhancer controlling the promoter. That means speed up reaction and slow down the reaction. And silencer always it is repress the mechanism. Then structural region. Structural
Synthesis.
region
Synthesis.
For amino acids, sir.
Synthesis. Yeah.
not amino acids, but here the synthesize the triplet codons. Synthesize the triplet codons on the DNA strand. As a result, due to the activity of structural genes, mRNA strand prepared. mRNA strand prepared due to the activity of structural gene. Okay, when these structural genes are activated, When they are activated, first the promoter, before activation of structural gene, promoter should be activated with help of enhancer.
Yes.
Then only these RNA polymerase move forward on the structural gene. Then immediately formed RNA stand on the
Nothing
DNA.
but elongation.
That is called elongation. Okay. After that, the termination and that process. Termination and the process. Right?
Yes,
Is
sir.
it okay?
Yes.
Any doubt in gene or gene structure? Eukaryotic genes. Promoter importance, enhancer importance, silencer, and structural importance. Is it clear?
Yes.
Okay. And here, gene amplification, loss, rearrangement, it is no need, not involved in protein synthesis, not involved in transcription. That's why this process, no need. The page number 69, 69, 70 matter, no need. Because this matter not involved in transcription. Okay. What is the question here? The only gene structure, eukaryotic gene organization. Eukaryotic gene organization means what are present in gene. Enhancer present, silencer present, promoter present, and structure present. That's enough. And promoter, the separate special sequence starter box and the CCAT box also. That's enough. Retroposons, immunoglobulins, there no need. Okay. And are here given RNA polymerase 1 and 2 and 3. I will explain later. That means the page, the unit number. These all polymerases again repeated in block 2, molecular biology 2. In molecular biology 2, the restriction endonucleases and modifying enzymes, there are polymerase chain reaction. In this, again, this about RNA polymerases and DNA polymerases. DNA and RNA polymerase again repeated. That's why I will complete it later. Okay?
Okay.
Yes, sir.
Can I move from next unit? Gene expression in eukaryote, 5th unit. 5th unit. Okay. Hello.
Yes, sir.
Right. All of you, please respond. If you not understand, again ask me.
Okay, sir.
Over here, Kavita Gnaneshwari, Kamrushan, Kavita TGT, Jyoti, Lalita. Are you in live? Swati, Nagaraju.
Yes, sir.
Right. Right, next one. Gene expression in eukaryote, already the given introduction yesterday. Now central dogma of molecular biology. The simple 5.3 central dogma, simple transcription, translation together called as central dogma of molecular biology. That means first one, DNA to RNA, RNA to protein, RNA to protein. A DNA to, again DNA also prepared by
Replication.
Replication. DNA to another stand, that is mRNA, tRNA, rRNA, that is called
Transcription.
called transcription. Transcription. RNA to protein, this process called a translation. This process called as translation, right? Transcription process takes place in eukaryotic, takes place in nucleus. And in eukaryotic, all enzymes, that mean all polymerases 1 and 2, 3, they are all present in nucleus. In prokaryote, this is occurs in cytoplasm because there is no nucleus. There are no nuclear membrane. That's why this transcription translations occur in cytoplasm in prokaryote. But in eukaryotic, this transcription process, that means DNA to prepare RNA in nucleus. That is one step. After preparation, this RNA transferred to the cytoplasm, where translation takes place. As a result, protein stand prepared, where translation takes place, where the protein stand prepared, right? These proteins are made up of amino acids. I will tell you later how the amino acids are added. So this is the central dogma of molecular biology. In this first, let's complete about the transcription process in eukaryote. Later, translation process in eukaryote. Only they are asking about transcription process eukaryote only, not prokaryote. But anyway, I will add some prokaryotic character also for better understanding, right? Please join again in C8. After quit the class, again join in C8 link. Okay, that is that would be run from 3 to 4:30. Okay.
Okay, sir.
Please join again.